Hi, welcome to Neuroverse
A podcast hosted by Clara.
and Carolina, where we discuss all matters from neuroscience to philosophy and beyond.
Today's episode is on neurogenesis, also known as the growth of new neurons, and specifically whether it occurs in the adult human brain. And to explore this topic, we're joined with a special guest, Carlotta Barelli. Carlotta has just completed her master's in neuroscience at King's College London and is now starting a PhD at the Human Technopole Research Institute in Milan, where she will be studying cortical development. Hi, Carlotta. Thank you so much for joining us today. Would you like to explain why you're interested in neurogenesis?
Hi. Yeah, so I personally found neurogenesis always really interesting because it's all about the birth of neurons from a fully undifferentiated stem cell. And from there, you get like these beautiful neurons, and not only the neurons, but all of the cells that make up the nervous system. And when we talk about neurogenesis, we mainly talk about embryonic neurogenesis and what happens during development. But actually, there has been some recent studies, or not so recent, already 50 years ago, some people started claiming that neurogenesis might also happen in the adult brain, which of course was really interesting and went against the biggest dogmas in neuroscience, which was that nothing may be regenerated, but everything may die, which was already stated by Monica Hull. Yeah, so that led to a lot of interest in neurogenesis and if it actually happens in the adult, because it's still a really, really big controversy, especially in humans.
So, what type of research has shown that it actually does occur in humans?
So the research in humans, as I've said, is quite limited because to actually test that there is ongoing neurogenesis, we would need animal studies where the ethics, of course, are much more lenient, and we would be able to, for example, utilize radioactive substances. Actually, the first evidence of adult neurogenesis comes from these radioactive substances like VRDU. So these are thymidine analogs which can integrate into the DNA during its duplication, its synthesis. And then by looking at those cells which have this BRDU, you can see that the cell has regenerated. So that is a really clear indication of an adult-born neuron. But as I've said now, this cannot be done in humans. And another really interesting piece of evidence for adult human neurogenesis was based on carbon dating. And this was, again, like once off probably because this comes from nuclear bomb testing, which was carried out in the 60s. And basically, during those years, the carbon-14 levels in the atmosphere were really high. And these were actually able to integrate into the cellular genome. And thus, you can basically estimate the cell's age based on the levels of carbon-14 that it has inside its genome. And this showed again that some cells in the adult human brain were actually born after the person's birth date.
This method's super cool. Like, I would never expect birth dating to be done to study neurons or neurogenesis at all. So that was really interesting.
Yeah, definitely. It's really interesting, but again, like really limited, and probably will not be able to do that. Again, recent studies have tried to do some single cell sequencing on human tissue, which was resected from surgery. And this is in the hippocampus because basically in the adult human brain, there seems to be two major sites of neurogenesis, and one is in the hippocampus, in the dentate gyrus, and the other one is in the subventricular zone of the lateral ventricle. And so this recent study had some tissue which was resected from hippocampus, probably from surgeries of epileptic patients. And yeah, they carried out single cell sequencing to see whether some of those cells had signatures of stem cells. And they actually claimed they didn't. So again, huge controversy, and the field is just like going back and forth. Yeah.
So, what are the implications that neurogenesis does occur in the adult brain? Because as you mentioned previously, we thought that that was impossible, that once a cell would die, that would be it. But now there is evidence that there is birth of new neurons. So what are the implications in terms of cognition?
Yeah, that's really interesting. And I think a lot of the funding that goes into neurogenesis is because potentially this can have a huge impact on cognition and potentially in some of the therapies for neurodegenerative conditions. Of course, we're really, really far from that because if neurogenesis occurs in humans, first of all, it does occur at a really low level. So it's estimated to be like 700 neurons per day over the billions and billions of neurons that we have. So it's like really, really low turnover. But still, it seems to have some function. And a lot of this evidence comes from mice and rats, and their neurogenesis seems to really boost spatial learning, pattern separation, so trying to discriminate between two really different inputs and trying to generate different outputs. And in general, cognition seems to be boosted by neurogenesis and by the fact that these newly born neurons probably are much more plastic compared to the more mature neurons, and thus they're adding plasticity to the circuit and helping with the formation of new memories. Yeah, but again, this is all evidence from mice where a lot of the new neurons, at least the ones that are generated from the lateral ventricle, the subventricular zone, end up in the olfactory bulb in the nose. And we know that mice are really, really good at olfactory learning, and they use their olfaction a lot. And neurogenesis seems to help with olfactory learning and olfactory discrimination. But again, like the relevance to humans is quite limited, I think, because we don't rely on olfaction, and the neurons that are born from our lateral ventricle probably don't end up in the nose. And studies have shown that they may end up in the striatum. So again, really different.
I find it really interesting how these interspecies differences in neurogenesis are related to the functions. So as far as I'm aware, neurogenesis in the olfactory system doesn't really occur in humans, as you just said, but it does occur a lot in mice, whereas neurogenesis in humans is thought to mainly occur in the hippocampus. And this makes sense in terms of humans needing to fulfill their cognitive capacities. And as we evolve, we rely more and more on these cognitive capabilities, such as memory, to survive, rather than a mouse, which would rely more on their olfactory system. And so there's definitely this evolutionary link, I think, between neurogenesis across species. And what I find even more interesting related to this is that it's been shown that there's like a non-phylogenetic development of neurogenesis across species in terms of the fact that cognitively more complex animals don't necessarily have more neurogenesis. For example, dolphins are a really cool example of they're very smart animals, and they also have very large brains and a very long life, but neurogenesis has not really been shown in these animals.
Yes, I'm super passionate about this. And what I find the most interesting about this is that if we look at developmental neurogenesis, so the birth of neurons during development, it's kind of the opposite, because it's like the more advanced embryonic neurogens that we get, the more cognitively advanced the species are. So if we think about humans, they have like a really, really big neurogenetic capacity during embryonic development. They have different types of stem cells, different type of radial glia, giving birth to different types of neurons. And this is much, much simpler and much reduced in mice. But then if we look about adult neurogenesis, it may be kind of the opposite, as you said. And it could perhaps be that less cognitively advanced species have actually more neurogenesis. Yeah, so it is really interesting. I don't think that there's that much known about that.
It's interesting also how you pointed out that in mice, the neurogenesis has a greater effect on the olfactory bulb, whereas with humans, it's more in the striatum. And not only how the hippocampus is incredibly important for memory formation, but the striatum is also this really important center for decision making. And so the stream of information of the exterior world and the contextual information of your background, and how cognitively this is a really important function for humans. So it's interesting, and again, makes sense that with mice, it's in a different area of the brain. Also related to this phylogenetic separation of neurogenesis effects in terms of cognition, Gavin A. wrote this paper about adult neurogenesis, and he compared the types of memory in rats versus in mice, and that apparently there were studies that showed that after neurogenesis there was an increase in memory formation in mice, but that with rats it was the opposite. It caused memories to forget. It caused the mice to forget. And so I thought that was really interesting how, as you also mentioned in the beginning, a lot of controversies with neurogenesis, these two clearly contrasting studies about how in one it increases memory formation and how in the other it increases forgetting.
This also reminds me of something I came across. So neurogenesis is a form of plasticity, but a big debate in the neuroplasticity world is stability versus plasticity, because obviously we see plasticity as a positive thing because it enables cognitive flexibility and memory formation and learning, etc. But you also need to keep the network stable in order for it to function. So it's always sort of a trade-off between plasticity and stability in the brain to optimize functions such as learning and memory. And this was mentioned in the context of neurogenesis way back in 1985 in a paper by Pasco Rakic, where he explored the limits of neurogenesis in primates and suggested that neurogenesis may be limited to enable plasticity, but also stability. So in other words, you need just the right amount to serve these cognitive functions.
And I guess also related to this increase or decrease in plasticity, the fact that in this study it induced the forgetting of memories, that may not necessarily be a bad thing, because it could just be like a homeostatic plasticity of like washing out of unused memories. So it may not necessarily be a negative thing. It could actually end up enhancing the cognitive performance and function overall.
Yes. Yeah, no, related to that, I was just going to say that the potential functions of neurogenesis are so diverse, and people think that it's linked, for example, to infantile amnesia on the one hand, or on the coding of temporal correlative memory or pattern separation, spatial navigation. It's like so much going like all the way, as you said, from like forgetting to remembering. And in terms of the like homeostatic mechanism which might underlie neurogenesis, I think the balance between making new neurons but also maintaining stem cells is really important. And if you think about a human brain, first of all, we have a much more expanded lifespan compared to the mice, and we also have a much bigger brain. So I think it's quite clear that we cannot have perhaps the same rate of neurogenesis as a mouse, because our stem cells have to last for so long, and they have to be able to keep on producing neurons for the whole lifespan. And that's why the field really thinks that a lot of the cells in the adult human brain, the stem cells, are quiescent. And quiescence is a really big thing when we think about adult human neurogenesis.
Could you just explain a little bit of what quiescence is?
Yes. So basically, quiescence can be defined as the exit from the cell cycle. So it's stem cells that still have the capacity to reproduce themselves and produce new neurons, but they're basically stopped in the cell cycle. And a lot of different extrinsic and intrinsic factors can stimulate their cell cycle and reinitiate their reproduction, which also makes me think about like all of the factors that influence neurogenesis, because that's like another huge topic, and people claiming that running increases neurogenesis and learning and swimming and meditating and everything seems to increase neurogenesis, because again, like so many factors come into play when we think about stem cell behavior and control of cell cycle.
But also the integration of new neurons into circuits requires activity, and it requires their usage. So even if you cross that barrier of turning from a stem cell to a neuron, then you also need activity to occur to keep in the circuit and to integrate.
Yeah, that's very interesting. Never thought about that. But yeah, it may come afterwards. And yeah, studies have focused on looking at the amount of stem cells that have turned into neurons, but not that many studies actually look at the activity of these cells. Because yes, maybe neurons are made, but if they're not actually functional and integrated into the circuit, they're not really helpful there, I think.
Yeah, and I know, as I've done some research on the dentate gyrus, I know that newborn neurons have been shown to have different electrical activity to more mature neurons. And although the evidence is minimal, this is thought to play a role in, as you mentioned, pattern separation, in that mature neurons are less responsive. The hyperexcitability of newborn neurons has also been linked to the inhibitory circuits. So like inhibitory circuits may develop over time and inhibit mature neurons more, hence their lack of firing comparatively. But there's so many questions yet to be answered in that field.
It's true about the inhibitory neurons. Thinking about the olfactory bulb in mice, basically the majority of these neurons that are newborn, they become either GABAergic or dopaminergic inhibitory neurons. So perhaps the fact that they're more broadly tuned leads to more inhibition of the pyramidal neurons, and thus like more discrete firing of the pyramidal neurons. Yeah.
I wanted to mention about quiescence. I find quiescence really interesting because it's all about the potential of stem cells, of course, to turn into neurons. But this leads me to my biggest question for you, Carlotta, which is, does the brain have unlimited potential, or are we limited by the number of neurons and our lifespan? So for example, with quiescence, there's like a potential for these cells to turn into neurons later in life. But if we accomplish what seems to be a huge human goal of expanding our lifespan, how will this change our quiescent potential? And will our brains need to adapt to this?
Such a big question.
Yeah, it's a huge question. Definitely really interesting. I don't think I have the answer to this, but what I can say is that definitely aging seems to be a huge factor when it comes to neurogenesis and quiescence. And basically, the more we age, the less actively dividing stem cells we have. And this seems to come from inflammatory signals like higher levels of inflammation in the aging brain, and thus a reduced capacity of these stem cells to actually regenerate. They still seem to be there. Again, this is all like rodent evidence, but they are there. They're just quiescent. And really interesting experiments have taken blood plasma from young mice, and they've just basically infused the young plasma into an old mouse, and this led to higher levels of cyclin neuroprogenitors turning into neurons. So I think my answer to this really big question is that probably we do have potential. It's just like about activating that potential and like the signals that regulate it.
Right. So do you think, instead of further thinking about, does neurogenesis actually exist in adults, in humans, we should just keep studying the mechanisms of neurogenesis to see how we can harness this knowledge to promote it?
Yes, I think definitely looking at mechanism is always really important. But again, the tricky side of it is that looking at mechanism in a human is extremely difficult because no invasive experiments can be done. So we only have correlative evidence in humans from this really limited material. But yeah, definitely trying to study mechanism in a rodent is the way to go, I think.
I wanted to mention something which we actually were discussing earlier again, which is the timing of neurogenesis across life, just because I wanted to highlight a really good review paper written by Bond et al. Just this year, which is entitled, "What is the relationship between hippocampal neurogenesis across different stages of the lifespan?" And as you mentioned, Carlotta, there seems to be like two different types of neurogenesis, which is the initial short burst of neurogenesis in early development, and then a more prolonged low-level period of neurogenesis throughout the life. And something that I found really interesting that was mentioned in this paper was the link between neurogenesis and gestation time. So rodents have quite a short gestation time, and in this case, hippocampal neurogenesis mainly occurs during the end of gestation and then it peaks at birth. Whereas in humans, most neurogenesis begins in early gestation and then it peaks between the first and second trimesters. And so the low level prolonged neurogenesis actually begins like earlier in the gestation period compared to rodents. Well, I think this was an important point to emphasize because I like to view neurodevelopment as occurring throughout life. And neurogenesis is kind of a perfect example of this because, again, linking back to the first thing you said, Carlotta, that while people used to think of the brain as something that develops and then stays that way, but now it's much more widely believed that instead everything that occurs in the brain is a continuous process that occurs all throughout life.
Yes, definitely. I fully agree with that. And I think that we should not look at neurodevelopment just in terms of what happens during gestation and like the first few weeks after birth, but definitely is a lifelong process. And when we think about the continual regeneration of the nervous system, I think we should not be limited to thinking about neurogenesis in terms of birth of new neurons, because we know that a lot of different cell types exist in the brain. We have a lot of different glial cells, and these glia actually seem to have a much bigger regenerative capacity than neurons. For example, after a traumatic brain injury, we get microglia, astrocytes really proliferating and trying to perhaps help with the traumatic brain injury, but then also create a massive inflammatory scar. So yeah, I think like the process of neurodevelopment is definitely really vast and can be expanded to lifespan. And also, if we think about cancer again, cancer, according to some theories, cancer is the reactivation of these developmental pathways. So if we look at a cancer cell, it has hallmarks of a stem cell during development. So it's able to proliferate, to generate a lot of different neurons. It has less dependence on growth factors. Yeah.
And perhaps also linked to neurodegenerative disorders.
Yes, definitely. Like the link between neurogenesis and neurodegeneration is also being explored quite a lot, especially in the context of Alzheimer's disease, because a lot of rodent models of Alzheimer's disease seem to have decreased level of neurogenesis in most cases. But anyways, these alterations in neurogenesis seem to appear quite early on in the pathology. And this could be really useful in terms of biomarkers of Alzheimer's, because we know that it's really difficult to diagnose. But it seems to be really one of the first events occurring. And in terms of humans, some studies have shown that both the aged brain and brains from Alzheimer's disease patients have decreased levels of neurogenesis. And then if we think back about cognition and how neurogenesis helps with cognition and the formation of new memory and all of that, it would also make sense that a reduced level of neurogenesis is linked to a form of dementia.
Yeah, especially if neurogenesis is related to the role of pattern separation, which is a hallmark cognitive deficit of dementia or Alzheimer's disease, because it's the ability to categorize new memories.
Yeah, related to that, as we mentioned earlier, in terms of like there's a lot of activity and practices that we can take that potentially increase neurogenesis, such as the study that I read with the mice. They use the example of exercise. Would you say that it's a bit of a leap to interpret these studies that use examples such as exercise to say, okay, well, an increase in exercise means an increase in neurogenesis? Or how would you go about delivering the actual message of neurogenesis and how it's related to exercise or meditation or other things.
Yeah. No, I think we have to be really cautious because again, like the evidence is there, but it's just coming from rodents and it might play a really different function in humans. But yes, like intervention and like really easy interventions like running or meditation or singing are recommended by the Alzheimer's Society. And again, like these studies in rodents have shown that there's quite a direct relationship between these interventions, for example, running, and an increased neurogenesis, and downstream of that, an increased ability in spatial discrimination or pattern separation. And also, all of these interventions don't lead to any harm, I think. It's just like about increasing health. And again, I'd like to think about neurogenesis as probably a biomarker of overall health because evidence has shown that it is increased by good diet, by running, by all of these just like general healthy living practices.
I think in humans, the evidence showing a link between exercise and neurogenesis is quite indirect in that they've shown that it's using neuroimaging, which indicates the level of oxygen in the brain and supplied through the bloodstream. And they've shown that running can increase blood oxygen, and also that an increase in blood oxygen is linked to better memory or cognitive performance. So there's that indirect gap. So, but I think that if you can form a link between these interventions and the cognitive outputs, and at the same time study the mechanisms that underlie neurogenesis, then hopefully one day we'll be able to combine the two and have a better, fuller picture of what's happening.
Yes, no, I fully agree. I think neuroimaging, even though it is an indirect measurement of neurogenesis, for example, but it is probably one of the best modalities that we can use to measure this in humans. And then, as you said, go on to rodent models to really look at mechanism and at the causative relationship between what we've observed in humans. And yeah, they look a lot at hippocampal volume, which of course can be viewed as limited because it's just like looking at volume. It's like a direct measurement of neurogenesis, but again, a lot of these different interventions seem to increase the volume of the hippocampus in humans.
So on that note, we'll conclude today's episode. Thank you so much, Carlotta, for joining us and sharing your expertise on the topic.
Thank you for having me.
Yes, thank you for joining us, Carlotta. This has been amazing. And if you enjoyed this episode, feel free to share it on Twitter and tag us. And do sponsor us in Ko-Fi because we'll reinvest all the money back into improving the quality of the episodes. Thank you.